best practices in palm fruit. My name is Stephanie Gordon, and I'm the editor of Fruit and Vegetable Magazine. Before we begin, I'd like to take time to thank BASF for their support for this webinar on fungicide resistance management. I'm joined today by Katie Goldenhar, who is a pathologist for horticulture crops at OMAFRA, Ontario's Ministry of Agriculture, and Anne McRae, who is a technical services specialist at BASF. Today, during our free 60-minute session, Katie will start by broadly talking about fungicide resistance management in the first half, followed by Anne, who will talk briefly about the Cevya fungicide. At the 45-minute mark, we will start the Q&A period. If you have any questions throughout the presentation, please type them into the Questions tab found on the right-hand side of your screen. Without further ado, I'll let Katie take it away.
Thanks, Stephanie. So my name is Katie Goldenhar. I am the pathologist for horticulture crops with the Ontario Ministry of Agriculture, Food and Rural Affairs. And I really want to thank you all for joining me today. I know time is very valuable, so I really appreciate you taking the time out of your day to listen to me and talk about this topic. So I'm going to be talking about fungicide resistance management, and feel free to ask any questions throughout today's seminar, but also feel free to reach out to me at a later date if you want to learn more or talk more about fungicide resistance management. So I have focused on apples, but I feel a lot of these practices and managements are also valuable across all horticulture crops where fungicides are used. So I want to talk and start about the impact of plant pathogens, and I don't necessarily need to talk to this about with a bunch of farmers who know that pathogens need to be managed, but I like to mention as often as possible that these plant pathogens have severe impacts to crop yields, crop quality, and health of either humans or animals, and do need to be managed. Now, these management strategies can take different forms, whether they're through cultural, biological, physical, or chemical, but they do need to be managed. And some examples to go through that are very historical. If you've ever taken a plant pathology class, you know that the ergot of rye is one of the earliest human health impacts from a pathogen. And so what this fungus did was it produced this black structure in the head of the rye, which you can see on the top right picture there. And this is called the ergot. And in the original drawings of rye, they actually thought that this was a part of the plant because they were drawing it with that picture. And so what happened was when this fungus was put in the flour with the grain and people consumed it, there was this toxin that caused people to hallucinate. They thought their limbs were burning. It caused death in severe cases. And this pathogen is still around today and still needs to be managed, and is a good example of the impacts that plant pathogens can have on health. Another pretty severe case of impact of plant pathogen is late blight of potatoes and tomatoes. So the potatoes were the main source of food for the Irish in the mid-1800s, and what happened was this Oomycetes fungus came and wiped out their entire crop. It also rotted the tubers, and this fungus is still around in all major potato growing regions across the world and needs to be managed in order to maintain the yield and the quality of potatoes. Another one is Panama disease. This is a soil-borne disease that's fairly interesting. So what happened was there was a dominant banana cultivar. There wasn't much variation in the cultivars. And in the 1900s, this pathogen caused the industry to shift over to this more resistant cultivar because it wiped out that entire cultivar. So there are some severe impacts of pathogens that can occur. And so they do need to be managed, and fungicides is one of those management tools we have in a toolbox for disease management. So when we look at the development of early fungicides, they were multi-site, they were inorganic, and one of the earliest inventions or observations with fungicides was actually a scientist in the 1600s looked at treating cereal seed with brine, which is salt water, versus just water. And what they saw was that there was this reduction in the fungus growth that was on the cereal seed. And so that was one of the early examples of a fungicide before we really knew what was causing disease. And in the 1800s, one of the earlier fungicides that's still around in some form or another was the Bordeaux mixture, which is a mixture of lime and copper sulfate, was sprayed actually to deter humans on grape crops. And what happened when it was sprayed, they noticed that there was actually a reduction in grape downy mildew, which causes defoliation of the crop. And so that was one of the earliest fungicides that's still around today. And so before the 1940s, chemical disease control was mainly a mix of inorganic chemicals. And after the 40s, what we started to see was the development of most of our multi-site broad-spectrum fungicides that are still around today. So the ECBDs, which includes mancozeb, captan, and chlorothalonil. So those multi-sites were developed fairly early. And after the 60s is when we started to see the development of these highly active single-site fungicides. And so modern fungicides, as we know them today in these different groups, are highly specific and active at much lower rates than these inorganic multi-site chemicals that were originally used before the 40s. And so you can see here there's a few examples of some groups that are still used that were developed fairly recently. This was just 50 years ago, 20 years ago, 30 years ago, so they're still relatively new. And you can see this chart here. I think it's really interesting looking at the pounds of active ingredient that were used back in the 40s has significantly decreased with the use of these single-site fungicides because they're highly active and act on that fungus or pathogen in a certain way that makes them applicable at lower rates. And so when we're using fungicides, there can be many different reasons for fungicide failure. And fungicide resistance is not always the reason. And so when we're going through different reasons why the fungicide might not have had the efficacy that it was supposed to, there are different factors that can become at play. So the wrong product, so making sure that the proper pest has been diagnosed so that you're using the right product. Using the wrong rate can lead to a fungicide failure, so making sure that you know what rate to use and you're following the label. The improper timing, so you need to make sure that you know when to target a specific pest. Low water volume, so a lot of these fungicides are locally systemic or not systemic, and so making sure that you're having proper coverage of the crop in order to maintain that fungicide efficacy. Spray water at a different pH can have an effect on some fungicide efficacy. Sprayers not being calibrated properly can lead to fungicide failure. And then also poor weather conditions, so something like wind or rain, either during or immediately after a fungicide application, can lead to fungicide failure. Fungicide resistance can result in fungicide failure if all of these other factors have been properly followed. And so I'm going to be talking about fungicide resistance and understanding why it occurs and what it is, because I think that can really help in how we manage it. And so fungicide resistance is an acquired heritable reduction in a sensitivity of a pathogen to a specific fungicide. So it's less sensitive. It's not something that occurs as a one-off and goes away. It is something that can be passed down from that pathogen to that population. And field resistance is really when we start to notice it in a field. And so that's when the population of the pathogen, of the disease, is mostly made up of individuals that are less sensitive to the normal rate of a fungicide that was previously providing control. And so I think a topic when we talk about fungicide resistance that comes up is there's a question that do fungicides cause fungi to mutate or change from sensitive to resistant? And the answer is no, because it's a natural process of evolution that when these fungi are reproducing either sexually or asexually, they're mutating. And those mutations happen at a very small rate. So there's 1 in a billion or 1 in 100 billion. So they're happening at a very small rate. But what happens is this is more frequent. These mutations that can confer resistance happens more rapidly in some species due to the reproductive frequency. So you can imagine if you have a pathogen that's reproducing very, very frequently, that these mutations can occur at a higher rate. And so here's a diagram of a strawberry field. This was used by my predecessor, but I think it's really important to show how resistance can occur from just one isolate to across a field. And so you have a pathogen, that's the black dot on the strawberries here, and it is sensitive. It's the wild type. It's sensitive to a certain fungicide that's been tested and shown efficacy. But then you have the blue dot at the bottom, which is a mutant that is naturally mutated at a certain spot to confer resistance to a certain fungicide or group of fungicides. And so what happens as you spray that field with a certain fungicide is it will wipe out the sensitive population. It'll provide proper efficacy, but then that one spore or that one lesion that was able to confer resistance to that group is now able to reproduce and start to move across the field. And so what happens is if you're spraying the same product over and over, you're allowing for that population to continue to reproduce, continue to spread across the field. And so we'll talk more about management strategies to eliminate this factor, but this is how it starts to spread, is if you're using the same product, this resistant population or insensitive population is continued and allowed to spread across the field, becoming the predominant population. And so when we look at resistance and how it can occur and there are different risks that are associated with it, we can look at three different sides of a triangle. And so the one is disease, and the disease risk I've already touched on. So it's mainly associated with a certain pathogen and its reproductive frequency. And so weather factors can play into how frequently it's reproducing, if it's surviving, if it's sporulating well. But the disease risk is really associated to a specific pathogen. The fungicide risk is also associated to different groups of fungicides. So whether it's a systemic fungicide, it's a single site fungicide, it's a multi-site, all of those factors play into the fungicide risk on resistance. And so each fungicide group, known as the FRAC group, has a risk associated with it. And then the last piece of the triangle that I think is really something we can focus on is the agronomic risk. And so using practices that promote healthy plants and reducing the reliance on specific fungicides or using other factors such as genetic resistance or tolerance can really reduce the resistance risk when we're relying less on some of those other factors. And so I'm going to go through an example in apples as to what I mean by all three of these risks contributing to resistance. And so in apples, this is mainly based on Ontario. What we have is different diseases on the left-hand side and their associated resistance risks. These resistance risks are designated by FRAC based on that pathogen. And so you can see something like scab is at very high risk of developing resistance. Something like fire blight, powdery mildew is medium to high. Our collar rot, our bitter rot are medium, and black rot and fly speck sooty blotch are low. So that's just due to the nature of the pathogen. Then when we look at the fungicide group, so these have been grouped from the highest resistance to the lowest risk of resistance. And so what you can see is the commercial name on the left-hand side, the FRAC group, which you find associated on each of those labels, and the resistance risk on the right-hand side. And so your group one, so Senator, Murfect, are at a very high risk of resistance, as well as the group 11, so the strobilurins. Their antibiotic, streptomycin, is also at a high risk. And then as we move down, we get into the medium to high risk, which is your group 7s, your STHIs. Then we have another antibiotic is a medium risk. Group 3s, 9s are medium. Group 12 is a medium to low. And then our low risk materials are Aliette and Phostrol, so phosphorus acids, Allegro, and your group Ms are all at that low risk. And that's based on the molecular nature of the fungicide. And so when we put those together in a matrix, I'm going to walk through this. So I know it looks a bit complicated to start, but we have the diseases on the left-hand side. And the high-risk diseases are at the top, and they get a 3. So they have the highest number. And then it goes down from there. And then on the right-hand side, or on the bottom, we have the fungicides. And so the high-risk fungicides are on the right-hand side, and they go down to the low-risk fungicides here on the left. And so what you can see is when you are using a high-risk fungicide, like an 11, a 25, or 1, with a high-risk disease, you are getting a very high risk of resistance. And so these are the applications that are extremely important to follow resistance management practices. When you're using something like a M on a high-risk pathogen, you're still at a much lower risk of developing resistance. The risk is not gone, but it's at a much lower risk when you're using these low-risk materials. And so I mentioned the third risk, so the agronomic risk. And so agronomic risk is highly variable depending on location, season, your integrated pest management strategies. But I think this is a fairly good example for Ontario agriculture. And so your agronomic risk on some of your diseases are going to be higher than others. And a lot of it is how often you're spraying for those crops. And so spraying for those diseases. And so something like scab, where you're putting on 8, 10, 12 applications per season, is going to be at a higher agronomic risk of becoming resistant. And something like powdery mildew, where again you're relying on a lot of fungicides for control of that pathogen, is also at a high risk. Fire blight as well is at a high risk. Then we have a lot of lower risk where you're maybe spraying once per season, or you're not using fungicides for control of that disease. That is where you have lower risk of resistance as well. And again, this depends on the cultivar that you're using, but this is an example. And so when we put that back into this risk formula, agronomic risk is now here on the right-hand side. And you can see that now we have a very, very, very high risk of resistance for something like scab. When you're having a very high agronomic risk, you're using high-risk material, and you have a high chance of resistance already within that pathogen. And so that is something, especially in apples and pears, where resistance management strategies are really, really important when using some of these higher risk materials for developing resistance. And we know that there's resistance already to some of these. So what can resistance look like in a field? So there are two types of resistance as we want to talk about it. And one is called qualitative or single-step resistance. And this is when resistance occurs very quickly within a population. There's only one or maybe two genes that are mutating to confer resistance. And the difference between sensitivity and insensitivity is very clear. And this is also a very stable type of resistance. So the pathogen is resistant, and even if there's an absence of certain fungicides, it's still able to become the predominant population. So it's very hard to reintroduce those type of fungicides later on. So some examples are sugar beet across release spot to group 1s, potato and tomato late blight to phenylamine fungicides, so those are group 4s, apple scab to benylate. So those are some examples where it was very clear distinction between sensitive and insensitivity to those specific fungicides. And this is kind of what it looks like when we're looking at a chart. So you have the population, so the fungus group on the left-hand side, and then the shift between insensitivity and sensitivity on the bottom here. And so with this single step, what's happening is that population is very quickly going from sensitive to insensitive. So that's a very clear-cut type of resistance. The other type that we see is quantitative or multi-step resistance. And so this is something that gradually happens over several seasons. And this is when there's multiple minor genes that have been mutated to confer a partial decrease in sensitivity. And it's highly variable within a population. So it's not as clear-cut as the other type of resistance. And this population, when a pathogen has this type of resistance, has been shown to revert back to sensitive if fungicides have been discontinued. And this is likely because that resistant population has some sort of fitness penalty. So when it's in the absence of a fungicide that it is resistant to, that sensitive population is able to overcome that resistant population. And so some examples of that are apple scab to DMIs or group 3s, and apple scab to dodine as well has been an example of this type of resistance. And so this is what it looks like on that same graph. So you have the sensitive and insensitive on the bottom. And what you're seeing is a very slow progression from sensitive to insensitive. So it takes a lot longer, and that population shifts very, very slowly over to that side. And so some things that we talk about when we're talking about resistance is cross-resistance versus multiple resistance. So you might have heard cross-resistance when we're talking about the same group of fungicides. So on the figure here, we have apple scab isolates from Pennsylvania, And they've tested them for resistance against myclobutanol and fluconazole and difenoconazole. And so what they can see is that there is resistance to myclobutanol, there's resistance to fenbuconazole, and there's cross-resistance between those two. But what's interesting is that a fungicide, difenoconazole, which is also a group 3, does not have cross-resistance to these other group 3s. So there is the possibility for cross-resistance or not cross-resistance within the same groups. Now there is what's also known as multiple resistance, where a pathogen is able to become resistant to multiple groups. So that's what we call multiple resistance. And that is something that's happened with botrytis in strawberries, which is an example here. So that pathogen has been able to have resistance to multiple groups of fungicides. And so now we know some of the reasons why resistance can occur and the risks that are associated with it, but what can we do to prevent it? So reducing reliance on fungicide makes it so there's less selection pressure. So those fungus populations are going to be mutating and naturally conferring resistance. But if there's no selection pressure put on them by a fungicide, then there's less chance that that's going to become the predominant population. So using integrated pest management techniques such as resistance in cultivars or tolerance in cultivars, using models to begin applications, removing infected material, make sure that scouting is happening, and managing crop growth to optimize airflow, because a lot of these pathogens like really moist, wet conditions, and making sure that there's enough airflow can really reduce the pressure of a lot of these diseases. So when you are using a fungicide, because they are a very useful tool and sometimes one of the only tools against certain diseases, there are some steps that should be taken to make sure that they are managed to reduce the selection for resistance. And so one is rotating. So make sure that you know what frac groups you're applying on your crop and rotating between those, and making sure that those products have proven efficacy on the targeted disease. Another option is to tank mix with multiple modes of action on the same pest. So this can be done through a tank mix or through a premix, but making sure that on especially these high-risk pathogens like apple scab, that both of those products in that tank mix or premix are active on apple scab or on that high-risk disease. Using low-risk fungicides such as the multi-sites is also a really valuable tool to reduce the selection pressure on some of these single-site modes of action, and they can be used in tank mix or rotation. Limiting the number of applications per group per season. This varies based on the pest, it varies based on the pathogen, but in general, limiting the number of applications reduces the selection pressure for that certain group, and that can be done per season. Applying preventatively is also a really good best management practices in avoiding eradicant use. Some of the products have post-infection activity, but that is really hard to see when a fungus, the spore, it lands on a leaf and it starts to infect, but before it starts to produce spores. And so most of the time you can't see when that's happened. And so by the time that you're seeing a disease in your field, it's already been established. So you really want to try to apply preventatively wherever possible. Following application rates to ensure proper efficacy is really important, because you want to have individual, especially when you're putting tank mixes, you need to still follow those application rates, especially with these single site modes of action, because each of those fungicides needs to have individual independent activity on the pest in order to increase the probability of controlling those naturally mutant resistant isolates. And then also making sure that everything you're doing to apply that fungicide is done properly with the proper water volume, nozzles, pressure, is chosen properly, and it differs based on the stage and the pest. And again, that's because these fungicides normally have very little systemic movement within plants, and you want to make sure that you're applying it properly to your crop. So I want to quickly touch on the reevaluation of multi-site fungicides. I think it's a thing that's on everyone's mind. I'm not giving you any new information, but I think it's important to just touch on where we're at with some of these because it is going to be a challenge moving forward with a lot of these resistant management techniques without some of these uses. So something that's really important, I think, for palm growers is Captan, so it's a group M4. So the decision came out in 2018, the reevaluation decision, and all of the uses that came out in that decision need to be implemented by the suppliers as of 2020. And most crops do have a change to their use pattern, either through number of applications or re-entry intervals, and some uses have been canceled, so making sure that you're following the label of the product that you're buying. And here's just a quick snapshot of the changes that are happening to apples and pears. It's changing based on the density of the operation, but those re-entry intervals are increasing and the applications are changing as well. So making sure that you're knowing this year going in what the label says. Another group 4, this is full pit. This decision came out actually just a few weeks ago. Similar to other group M's, the main changes include increased re-entry intervals and the reduction of application in some crops, and then also a change in formulation to one of the formulations. And these label changes are coming into effect till 2022, so there's some time to review that and make those changes. Group M3, so this is Mancozeb, so this is another multi-site fungicide. That review is ongoing. The proposed cancellation of all uses was what came out. However, there was a response, and there is some cautious optimism that some uses will be remained, but we'll find out in June of 2020. That's their proposed final decision date, so we'll keep an eye out for that. Some other group M3s that were reevaluated in 2018 include Thiram, Furabram, Xyram, and Metram. And that decision came out in 2018. So again, uses canceled in 2020. Apologies, that should say all uses canceled in apples for 2020. And some uses in other crops, if you're growing other crops, have been retained for Thiram and Metram. However, you need to make sure that you're consulting your supplier about those. Then group M5, chlorothalonil, not important for palm growers, but important for many other horticulture crops. That decision came out in 2018. So again, 2 years later, 2020, May 10th, all products sold by then will have the new label, which has key changes to the number of applications and specific re-entry timings for that product. And so I've talked a lot about fungicide resistance. You're welcome to contact me anytime you have a question about it, but there are some other really great resources out there. So Manage Resistance Now is a newer one that was put out by CropLife that has some really good information and case studies on growers who have practically implemented a lot of these strategies, both in weeds, diseases, and insects. So I highly encourage everyone to check that out. For the fruit crop and vegetable crop protection guide, at least here in Ontario, making sure that you're consulting all the newest versions. They have most of the label changes where possible. And then they also provide some information on the relative efficacy of a lot of these on pests. And then there's some other more technical guides if you're more interested in how fungicide resistance occurs. So with that, I'd like to thank you for your time. And that is my information there. Again, you're welcome to contact me anytime. And right now I'm going to hand it over to Anne. And bear with us, we just have to change over the presentation.
Thank you. Okay. Hi, everyone. As Stephanie said at the beginning, my name is Anne McRae. I work as a technical specialist in horticulture for BASF, and I wanted to take a little bit of your time to talk about our new fungicide. We will have registered. It is registered now this year, and for those in Ontario, we are just waiting on Ontario scheduling classification, but we expect that this month and will be for sale this year. So Cevya is a group 3 fungicide. Its active ingredient is trademarked as Revysol, but the technical name is mefentrifluconazole. It is, as I said, now registered in Canada and also in the US. US received registration last summer. And we have registered this not only on apples, but grapes, potatoes, stone fruit, sugar beets, peanuts, and tree nuts. So there are three key features of Cevya that I wanted to sort of touch on. The first one is the fast and continuous control of key diseases in apples and other crops. What we mean by fast is it moves into the leaf quickly, and then continuous is kind of a slow movement through the leaf to protect it. So first of all, we looked at a study sort of looking at that rapid uptake. So this graph shows uptake and translocation data of Cevya into the leaf, seeing that Cevya moves into the leaf quickly. Within the first 5 hours, you already have 50% of that product absorbed by the leaf and protected, so you also get a very quick rain fast. So it has a 1 hour rain fast, or essentially once it dries onto the leaf. The next is that sort of continuous release. So we took a look at what Cevya would look like inside the leaf. It kind of creates these reservoirs, and then they sort of move slowly up the leaf. So it is sort of systemic, but moves up through the xylem, so it won't move back, but moves up. So coverage is important, but it does have some movement, and it's slow movement, so you don't lose a lot of that product out to the tip of the leaf. We get sort of a longer duration of control. And this kind of graph just kind of shows how that works, and showing the green line is Cevya, and over 160 hours, we're seeing that it has this, it's still providing a lot of efficacy and has this sort of slow release process. The second sort of key feature is something that Katie actually touched on a little bit, is talking about this preventative and post-infection control. So we will always recommend, obviously, preventative applications of Cevya, because they are the best way to prevent the disease. But we all know that sometimes the weather doesn't cooperate, and sometimes, you know, it's sort of unsuitable to spray. So Cevya can provide you a little bit of post-infection control, but I do want to sort of talk about what we mean by post-infection. So this graph kind of shows you, so for apple scab, for example, you'll have a spore release. That spore will land on the leaf. Then it takes time for the spore to sort of penetrate cell walls, and then it colonizes, and then you get this pre-sporulation and then sporulation. So when those spores release and start spreading again. So all of this spore germination to pre-sporulation can take place, but you may not actually see that. So when we talk about post-infection, it's generally after a few days, or up to 4 or 5 days, that you have the pathogen present, but you are not aware of that. So when you look at sort of a breakdown of some of the different single mode of action products we have there, like group 11s, 7s, and group 3s, they all act slightly differently. So it's always important that any of them can be used preventatively, and we will always recommend that. But DMIs, or group 3s, like Cevya, do give you sort of a bit of kickback or reach back, just because they also work well once that spore is sort of in that colonization stage. So they may give you a few more days than, say, group 11 or group 7. But again, they also will work preventatively, so that would be your first option. So the last, and certainly not least, of our key features is something that I wanted to talk to you, or that I'm kind of excited about, is this unique new binding activity. And it kind of describes how Cevya is just slightly different than your traditional group 3. So as Katie kind of mentioned, she was touching on all of the different FRAC groups, and Cevya is designated as a group 3, which we also call triazoles or azoles or DMIs. And they're called DMIs because they are dimethylase inhibitors. So any group 3 product that you have works like this. It will attack the C14 dimethylase enzyme, which causes a breakdown of cell walls, which is what kills the fungus. So it's very important to understand that it's a single mode. That's the single mode of action. It inserts itself in the C14 dimethylase, and that stops this production of sterols that build up the cell wall, and that's how it dies. With Cevya, it is unique because it has these two sort of groups. So hopefully everybody can see my cursor here, but this part at the end is the azole group. This is what makes it the group 3. This part here, the sort of orange part, is an isopropanol unit. And having the two of them attached like this means that it has a lot better movement than your traditional molecule. So as you can see, it kind of is able to create this hook movement, or hook sort of formation to hook into the enzyme. And what that does is kind of gives it like a lock and key, sort of, to give an example. So traditional group 3s, it would be like if you had a key and you put it in the hole and it fit and it attaches, but you don't move it, so then it could slide out easily. And so it's maybe developed some sort of, it kind of attaches and unattaches. With Cevya, because it has this sort of hook movement, it's like putting a key in a hole and turning it so that it can't slide out anymore. So with studies, we've found that it holds on up to 100 times tighter or better than sort of your traditional group 3s, just because of this flexibility in the molecule. And this slide kind of shows how that works, or the studies that we did to kind of prove that, I guess you could say. So you're looking here at a graph. Along the bottom, we have DMIs, which are your group 3s. So 9 different DMIs that were taken from products that are used in Canada and the US and various other countries. And then we looked at multiple strains, like 7 different strains of key disease that shows resistance to group 3s. And so as you can see, for all of the different DMIs, we have various different control and resistance depending on where these samples were taken from. But with Cevya, because of this unique binding and hook, that it hooks into that enzyme much stronger, for any of the strains that we looked at, it showed really excellent control. So that's kind of how we found this worked in the lab, but then obviously you have to take it out of the lab and prove that in the field. So when we look specifically at apples, this is kind of the key features looking at apples. Cevya will be registered on apple scab and powdery mildew. Powdery mildew, at that rate range, we get suppression, but if you use that high rate, the 375 milliliters per hectare, you will get control, but this is definitely a superior apple scab product, so I'll kind of look at that mostly. When we looked at some trials, both in Ontario and Quebec and BC, we saw very good efficacy, even under really extreme pressures of apple scab, and we were comparing it, in this case, to Circatus, which is another of BASF's products, both working really well, but yeah, just showing really good efficacy. These trials were done as a single mode of action, so the Cevya and Circatus were used on their own, but of course, because this is just a group 3, we will always recommend that it be tank mixed with something like a group M or any other fungicide that also has activity on scab. So I often get, when we have new products, we get lots of questions about what it looks like and things like that. So these are some sort of like those frequently asked questions. It is in a liquid formulation. It's an SC. So you'll see in the picture there, sort of a thick white liquid. The package size will be, in one case, you'll have 2 4-liter jugs. Cevya, or the active ingredient Revysol, is now registered in many countries like the US, Mexico, Brazil, and Australia. So there are MRLs established in those countries. And then other countries, we will focus on getting MRLs established for those in the next few years. As far as tank mixes go, we are not aware of any watchouts for Cevya. It should tank mix well with any of your products, but because we can't test everything, we always recommend that you do a jar test first to make sure you're not going to get any sediment in your tank. And if you're worried about, multiple products in your tank, spray a small area to make sure everything is working properly. Surfactants and adjuvants have been used with this product. They do not need to be, but they can improve coverage, which can give you a little better efficacy. So there's no issues if you like to use a surfactant or adjuvant. And then, as I said before, this is a group 3 single mode of action. And so it is always recommended for resistance management to tank mix this with something else that is also active on whatever diseases that you're fighting. So if you do have any questions on Cevya or want to look up any of BASF's other apple products or any other horticulture products, you can check out our website, agsolutions.ca/horticulture, or send me an email. I am happy to answer any questions about that. I encourage you this summer to give Cevya a try. It's a really strong group 3 for scab, and we're focusing a lot of our scab control on group 7, so now it gives you something to rotate with, which is really nice. So I'm going to turn it back to Stephanie for final remarks, but thanks everyone for listening in.
Okay, awesome. Thank you so much to Katie and Anne. It was a fantastic presentation with a lot of good information. And so they will now answer your questions. A lot of questions have come in already, but if you have a question that you want to ask, feel free to type it into the questions tab found on the right-hand side of your screen. Before we begin the Q&A session, I would just like to take the time to once again thank BASF for sponsoring the session. And let's get to the questions. So the first question is for Katie, and someone is asking for clarification in the earlier part of your presentation when you were talking about resistance risks, disease risks. You also mentioned agronomic risk. Can you expand a bit more about what you meant in that context?
Yeah, so I think what we're really looking at there is a risk that resistance could happen to specific groups of fungicide, a specific fungicide in a specific crop to a specific disease. And there are lots of different factors that go into why that's going to happen. And so that's what that is getting at. There's factors that come specifically from the disease. So some diseases are naturally more prone to have mutants that can confer resistance, and that's a lot of times due to the reproductive frequency of those diseases. Then the other one is fungicides. So we have these different frat groups of fungicides which work on the pathogens, the diseases in different ways. And so some of those ways that they're working on those diseases, those pathogens are easily able to mutate specific parts and overcome resistance to specific fungicides, or become resistant to specific fungicides. And so that is a factor of the fungicide. And so that is where we get into the low risk materials versus the high risk materials. And then the agronomic risk is kind of what you can do to promote healthy plants. And so healthy plants have they have a natural defense system to some diseases, to a lot of diseases. And so the better you can do to have a healthy crop, the more you can reduce the overall risk. And even if those risks from those fungicides and diseases are still there, you can control that one side. You can control that by choosing the fungicide so that you're controlling that side of the triangle. But the disease is pretty hard to control because it's normally around and affected more by weather than anything that we can specifically do. So I hope that kind of clears things up a little bit.
Mm-hmm. So agronomic risk is just making sure that you have healthy plants so they are better equipped when disease pressure comes by, and that they get off to a good start essentially.
Yeah, for sure.
Okay, and so the next question we have is someone is asking for clarification on what the group numbers represent. You mentioned that Cevya is group 3, there's group M. Can you, either Katie or Anne, clarify what you meant by the group numbers?
Yeah, so group numbers, sorry, I should have better clarified. So the Fungicide Resistance Action Committee is a group called, we typically short form to FRAC. And so each active ingredient that's in a fungicide or that is the fungicide has a specific group that it belongs to. And so some fungicides and modes of action that are the active ingredients within the fungicides belong to the same group. And so that's where you have these different group 3s. So they're different products, they're different fungicides, but they all are in the same group. And so they behave on the same part of the pathogen. And so you have group 7s, group 11s, and so those all behave on different processes that go on in that pathogen to be able to make them effective in either stopping or killing that pathogen. And so those are what we call groups, fungicide groups, and those are designated by the Fungicide Resistance Action Committee. Hope that cleared this up.
Yes, and if you have any follow-up questions, you can always answer them in the right-hand side. So I'm going to jump ahead. This question is related to what you were talking about with groups. So someone is asking, is Cevya more effective than, and I apologize for my pronunciation of this, Fungicide-Azole? Either way, so some of you, I think it's marketed as Score, that might be its trade name, but it is another group 3 fungicide. Did you guys do testing on whether or not it was more effective than that particular group 3?
Yeah, I think, yeah, difenconazole, I think, is a group 3. Yeah, so what we found in the testing is that it binds more tightly than difenconazole. That was one of the products that we looked at. That doesn't mean that, like, so some of it does, because we looked at so many different strains of things, and sometimes it still works and sometimes it doesn't. We were just specifically looking at how well it binds, and so that's why we found that it's better. You know, there's no cross resistance, as was talked about, Katie talked about before. So I don't know if that answers the question, but it's also important, no matter which groups you're using, even with our new Cevyas, that we're still tank mixing it with another group so that we protect what we have currently. So that's probably the most important part.
Okay, and so the next question is actually about another fungicide. Someone is asking, why isn't BASF bringing diethanol fungicide to Canada?
So I think, so everybody knows, I believe they're talking about, there is another group M fungicide that is used in Europe that is not registered here. And a lot of the same sort of, I guess, reasons that the group M products are being limited here would still apply to that group M, even though it is a different product. So it is on our radar, and we know that it is available, but we would still run into, when we would have to have that approved by McRae, we'd kind of hit those snags. So it's not that we aren't looking at it, it's just that we have to be careful of, putting in all of the money and effort of that to just lead to the same conclusion that we came to with Metaram. So hopefully that helps.
And so the next question, and I believe this could have been for Katie's presentation, but someone is asking, why did you imply that low volume always results in low coverage?
That's a good question, and I don't necessarily think that it always does, but I think that making sure that you know what kind of coverage you're getting is really important, and typically, a lot of times that is associated with higher water volume. Now, there is a limit, right? You can't go too high or you're going to get runoff, but typically there is more coverage when you're using the appropriate water volume versus a really low water volume, and so that's where I was going with that.
Okay, I hope that answers that person's question. So the next question is for Anne. Does Cevya protect leaves that are unfolding?
So it does, yeah, I think they mean sort of as the leaf grows, will it move? It will still, it will move through the leaf, but it only has, we recommend a spray interval of 7 to 14 days. So it's not, present forever, I guess you could say. So it does, because it creates this reservoirs and moves through the leaf, then yeah, for those 7 to 14 days, then it will protect. But it doesn't move backwards. So it's just moving up through the xylem. So you have to be aware that that new growth, that it may still need to be protected.
Okay, and this is also for you, Anne, as well. So does Cevya effectively prevent spore germination?
As in, so if you use the product, I think maybe I'm not answering this question, but I'll say, so Cevya should be either used preventively or as a like post-infection, like 2 to 3 days after infection, if you know when your infection point was. So when the spores kind of penetrate through the cell wall and as they're colonizing, that's when your group 3s or Cevyas can still attack that pathogen and kill it. Mm-hmm. But after that point, then no, you won't. So if you see the disease on the leaf, it is not going to burn it off or get rid of it, if that's what they're looking for.
I think the question was, so it was, does it effectively prevent spore germination? And they asked, if it doesn't, why would you preventatively spray? So I think they were wondering if it's only effective, if it's not preventing the germination of the spore, why are you spraying preventatively, like before? Why won't you just spray, like you said, when you see it on the leaves?
Yeah, so you shouldn't, if you already see the disease on the leaf, it's too late for any of the systemic products. You spray it preventively because the product will move into the leaf and will protect it for those 14 days that it's there. So once it's there, then it's going to attack those spores, whether they've, when they land and if they colonize and anything like that. So that's why preventative is important and is also sometimes easier because it can be very hard to predict when there is, a spore event or to know when those spores have landed and where they have landed. So that's why I would recommend preventative. Once you've sprayed it on, it moves into the leaf and then it's there protecting.
Mm-hmm. I hope that answers that person's question. The next question is, what fungicide do you recommend to prevent fire blight? This could be for Katie or Anne, yeah.
Yeah. So fire blight is caused by a bacteria. So there are some antibiotics that are registered, but I'm not prepared at this point to make recommendations on those applications, but feel free to reach out to myself afterwards and I can follow up with some management for that.
Yeah, and so BASF has a product called Apogee, which is a plant growth regulator, not a fungicide, but we have shown really good activity on fire blight, but when it's used in a program, with like your streptomycin and things like that. So that is also another option, but yeah, it's best fire blight is a very difficult disease to control and you really have to look at it as a program approach as opposed to just like spray it once and it's fixed. So, yeah.
Mm-hmm. Okay, and so someone is asking if Cevya is registered for grapes in Ontario.
Yes, it is. It is registered for grapes for powdery mildew. Also an excellent powdery mildew product. Yeah.
Nice. Someone is asking what products are available for organic growers, and this could be for both of you, or maybe Katie might want to answer this question.
Yeah, so it depends on what pests you're looking at. There are definitely organic products that are registered across Canada and in Ontario, and our crop protection guides do include those organic products and make mention if they are certified through the organic body.
And there was a mention that I guess how you apply the fungicide matters. For Cevya, are there any equipment concerns or anything new, anything to be noted for calibration or spraying, anything like that?
No, it should be, I mean, you can use it just as you'd use any of your traditional group 3s or 7s or 11s, or the way you're spraying. There's no watchouts, or whatever standard equipment that you're already using should be fine.
And this is a question for Katie. Are there any red flags to be looking for when you are scouting your field that are, you know, red flags of resistance that you can keep an eye out for in your field? I know you had that demonstration where you had the strawberry field where you start to see low efficacy on one side of the field. Can you maybe like list off some things that growers should be mindful of when they are walking their fields?
Yeah, so I think it depends on the crop and on the pest, but being aware of those high risk diseases in your crops and in your area is really one thing to look out for, because you might want to look out for those escapes after a fungicide spray. All of those things I mentioned in fungicide failure at the beginning should be make sure that those things are all followed properly, and always you can bring out to myself or some of my colleagues if you are concerned, and then those samples can also be sent to lab and we can do further testing as well with some of those potential escapes or concerns of resistant diseases.
Okay, and this is another question for Anne. Will Cevya be registered on berry crops? And is it effective against Collotrium? Sorry.
Yes, so we don't have berry crops on the label currently. We are looking at a sort of wave 2 crops that we would like to add to the label, or to go through, we do sometimes get crops added on through minor use. So both of those options we're looking at for berries. So that would probably take a few years, but I'm hoping that we would have both blueberries and strawberries on the label. I'm not sure yet, because we still have to do more of the testing and look at the data for, efficacy for all of the diseases. But maybe stay tuned and we can have more information for you in future.
Okay, and just to be clear, on your slides, was that Ontario focused, or was that also for all Canada, other provinces?
That's
or
all.
What it's registered for? Okay.
Yes, that's Canada.
Okay, and so we are running short on time, so I'm just going to ask one more question, but both Katie and Anne can be reached out after the webinar if you have any follow-up questions. So the last question is, on grapes, what is the re-entry period and pre-harvest period?
Oh, for Cevya?
I believe so.
So re-entry interval is 12 hours, unless you are doing like girdling and turning and things like that, then it is excessively longer. I think around 30 or 40 days. I will have to double check the pre-harvest interval, but I believe it's 14 days.
Now, is that written on the label, or where can they go to double check that?
If they go on to our website, like agsolutions.ca/horticulture, you can look up the label, Cevya label. Just look under horticulture products and it'll have Cevya label there.
Okay, awesome. So I'd just like to thank Katie and Anne for their time, and be sure to keep an eye out for the follow-up email, which would include a recording of this webinar, and the webinar will be posted on our website as well. So thank you to everyone for tuning in. Thanks again to our wonderful speakers, Katie and Anne, and thank you to BASF for their support for this webinar.